I'd like to talk to you today about induced pluripotent stem cells and their potential use in psychiatry research. And basically what I want to tell you is that I think these cells really open new opportunities in psychiatry research for modeling diseases and for drug discovery studies. So the way iPS cells I think are going to be used primarily is in cellular assays for these disorders. And one has to start by saying that cellular assays in psychiatry, of course, aren't new. We're very familiar with using animal cells, so typically primary cells from rodents in modeling disorders and in drug discovery. We use heterologous systems like HEC293 cells that have been transduced with drug targets. We use human cells on occasions, either primary human cells, not very frequently, obviously, and also cell lines derived from humans. And more recently, I think people have used neural stem cell lines. In fact, we ourselves in some of our research have used human neural stem cells to differentiate into neurons and glia and use those as in cellular assays in psychiatry. But I think iPS cells really open up a completely new avenue for research in this area. And I think that's because of two really primary seminal properties. So one is that we can take these cells really directly from a patient. So we really are studying a patient's own cells, and that's pretty much unique, not entirely unique. There are lymphoblastoid lines that we're familiar with. But having the patient's own cells that we can actually turn into neurons and glia is really a unique advantage. And then the second thing is that these iPS cells are truly pluripotent, and that opens opportunities that I'll come on to as we go through this. So let's start by looking at iPS cells and where they come from and how you make them. So the thing about iPS cells is that you start actually with a biopsy actually from the patient. So this slide shows how we do it in our lab. There are variants, but this is our way of doing it. We start from a hair. So literally we can start with a single scalp hair plucked from a patient. Now, using technology that's been around for a long time, you can grow cells from that hair keratinocytes. But then here's the really key bit, the really magic bit, and that is that we can actually now, by introducing just 4 genes, just 4 factors into those cells, we can turn those cells into these iPS cells, these pluripotent cells. And this really is the groundbreaking technology for which Shinya Yamanaka won the Nobel Prize in 2012. And the point is quite simply this, that the iPS clones that emerge from this reprogramming process really are pluripotent. They're pluripotent in exactly the same way as the inner cell mast cells are in embryonic development. So inner cell mast cells are the only normal cells that ever carry this pluripotent phenotype, and they carry it just transiently. It's just this ephemeral property. And what really Yamanaka has worked out here with this iPS technology is a way of generating cell lines that have this iPS property, have this pluripotent property, really ad infinitum permanently. And then that allows us then to really do some really remarkable things, and we can turn those cells into really any cell type in the body. So that's what we mean by pluripotent. These cells have the potential to generate any cell type that makes up the body. And our colleagues in other disciplines are turning them into cardiomyocytes and blood cells and varieties of other cells. For us, of course, the key is that we can turn them into human neural cells, so we can turn them into neurons and glia. So we can do that really in 2 steps. Firstly, we can generate neural stem cells from these iPS cells, and then we can differentiate the neural stem cells and generate neurons and glia. And so here's the key. We've now got neurons growing in a dish that are actually directly derived from the patient's own samples. And recall that the disorders that we're often interested in, diseases like schizophrenia and autism and bipolar disorder, are highly heritable disorders, where we think that the real key to the etiology of the disease is carried genetically. So what we've managed to do is capture precisely the patient's genotype that has given rise to this disorder. And we've now got the patient's own cells growing in a dish with precisely that genotype. And so we can follow those cells as they differentiate and ask the very fundamental questions we'd like to ask, namely, what is it that goes wrong? What is it that's disturbed about the neural development of cells with this genotype that brings about the disorders in which we're interested? And really that's the primary key, that we're able to use the patient's own cells to model the patient's own disease. So this diagram just gives you some idea of what this kind of looks like. So we start off with the cells at the left-hand end of this picture. We use a procedure that we call dual SMAD inhibition that neuralizes the cells. And over the next week or 2 weeks, you get these cells that form these neural rosettes, as we call them, which is the first picture in the middle of your screen there. And that sort of flower-shaped structure is really the attempt of these cells as they neuralize to form something that looks like a neural tube. And in 2 dimensions, this neural tube ends up with these flower-shaped structures. And then over the next weeks and months in culture, we can differentiate those cells. And you can see in the picture on the right there, neurons derived from the iPS cells. So exactly the kind of human neurons that we were talking about. So I said 2 properties. So the first is that these cells capture the genotype actually of the patient. So you're actually working with the patient's own cells. But the second important property that I think is going to be really crucial in the use of these cells in psychiatry research is these cells, because they're truly pluripotent, they have a power of histogenesis. So what do I mean by that? Well, we're used to working with neural stem cells that have the potential to make neurons in glia. So they're multipotential. But they don't really build tissue. What iPS cells seem to have is really the capacity to really go through a histogenic process as if they were truly building tissue. So let me give you an example of that as shown on this slide when we're looking at the example in corticogenesis. Right. Now, if you look at the bottom right-hand picture, this is simply a histological section of human cerebral cortex, and it reminds us of something that's really fundamental about cortex, namely that it's a laminated structure. So you can see the laminae in this structure. And we know this is important functionally and structurally within the cerebral cortex. We also know that it's pivotal developmentally, because we know that these laminae are formed in a very specific order, and that ordered generation of cortical laminae is really fundamental in the process of histogenesis in the cerebral cortex. So this property of lamination is important not just in the adult, but also developmentally, and that's shown in the top right-hand picture there. And what this shows is that successive layers of neurons are generated, starting with the deepest first, and successive layers forming more superficially. So the first neurons to be generated are the neurons shown in red in this diagram. And then successive waves of neurons form more superficially, giving this inside-out developmental pattern of the cerebral cortex. Now, the exciting thing is that this also happens in these iPS cells, and that's what's shown in the data on the left. So this is data from our lab, but it really builds on the observations made by Pierre van der Heeg and Rick Livesey. And what it shows is that just as successive waves of neurons are generated in vivo, starting with the deepest cells, the same is true in these iPS-derived cultures. So the neurons are again generated whereby the deepest cells are generated first, and then more successive waves are generated later. So this potential for histogenesis, I think, is really going to be fundamental in our studies going forward. Let me give you a really significant example. So this is taken from the work of Dan Geschwin and Ricardo Dolmetsch. And this is a study using iPS cells to study Timothy syndrome. Now, Timothy syndrome is a complex disorder, but it's also a syndromic form of autism. And we know that it's generated by mutations in the CACNA1C calcium channel subunit protein. And that's shown in the diagram on the right. Now, when you look at neurons derived from iPS cells taken from Timothy syndrome patients, they have a calcium deficit. And the data shown on the left there shows a broadening of the action potential, precisely as you might imagine with cells carrying this specific mutation. And that's very, very encouraging because that's what we expected to see. And now you can use these cells in conventional kind of assays to go on and study the pharmacology and the physiology of this deficit. But more exciting to me is the fact that these cells also show a corticogenesis deficit. So if we look at the generation of these waves of cortical neurons, these deep layered cells followed by the more superficial layered cells that we were just discussing, what we find is these cells make fewer of the deep layer cells. More than that, not just fewer of the cells, but fewer of the type of cells that project callosally. So during development, the deep layer cells, these neurons that are going to project out to the cortex, basically have a binary choice. They can grow an axon that turns left and goes off and forms the callosal pathway, or they can grow an axon that turns right, projects through the internal capsule to subcortical targets. Now we know that the callosal projection is totally dependent on the expression of a gene called SAPB2. And we've known for quite a long while that if you mutate that SAPB2, you affect the ability of these cells to form these callosal connections. Now it turns out in these Timothy syndrome cells, there's a reduction in the number of neurons that express SAPB2 and a reduction of the cells that take on this callosal phenotype. So this really quite sophisticated property of corticogenesis has been captured in these cells in a way that you might imagine was impossible in single cell cultures and where one would have had to have looked at animal models to resolve. So I think this really is an exciting way forward for research in this area. So let me summarize what I've told you so far. So there are pros to iPS cells for future and psychiatric research. These are humans. The human cells, so they really are capturing human neural development. Secondly, they're an alternative to animal models. They won't displace animal models, but certainly they're a cheaper and more tractable and easily accessible way of studying disease than having to use a whole animal with all the ethical and cost implications that that has. These cells capture the genotype of the diseases we're interested in. They're robust, they're reproducible, they're tractable, they're manipulable both genetically and in terms of treating with drugs and other environmental factors. And they're certainly suitable for high content screening and therefore drug discovery assays. Now there are downsides. The first and obvious one is that the cells are really slow in their development. That is a reflection of their human nature. Human neural development is slow. Also, they don't mimic the environmental component of the disease. And it's questionable whether they can capture the system properties associated with disease. And certainly they don't catch the behavior that we're interested in. But nonetheless, I think they are an exciting new opportunity to look at cellular assays in the development and the study of the etiology of psychiatric disorders.